Hi, it's Jamie Batts, your instructor for Bio 221. This is Chapter 25, and this is going to be Part 3. If your mind is spinning because you just watched the Part 2 video and you're thinking, carbon schmarbin, why are we talking about 2 carbon, 3 carbon, 4 carbon, oh my gosh, I don't know about these carbons, then go ahead and look at page 924 and 925, and you'll think my discussion of carbons is much more simple. Because what I did was I basically simplified the entire process for you in a nutshell. So if it doesn't make sense, read the chapter, go back and re-listen to the video, because it's an uber simplified big picture of what's going on here in carbohydrate metabolism. So again, just to reiterate, the carbon comes in, creates pyruvate. Pyruvate goes through a series of steps, losing some carbons along the way, ultimately going through this cycle where acetyl-CoA, which is the former pyruvate, joins with oxaloxylic acid and makes citric acid, goes through this whole cycle, forms oxaloxylic acid again, joins with another acetyl-CoA, etc., etc. This cycle keeps repeating itself, yielding NADH and FADH2. So our next question is really, what happens to the NADH and FADH2? And that is really the last step of carbohydrate metabolism. Remember, the whole purpose of this process is to yield ATP energy, right? The energy currency for all of our cells. Well, so far, from this one glucose molecule, we've only gotten 2 from glycolysis. We used 2 in glycolysis, we made 4, so we're only yielding 2 ATP molecules at this place, or this point, I should say. The citric acid cycle isn't making ATP for us. It's making NADH and FADH2. So we're thinking, well, where the heck does this ATP come from? And that's the electron transport chain and oxidative phosphorylation, the last little mini steps of this process. And there's really no, I'm looking at your book right now, there's really no good diagram of this. It's kind of a crazy process. I guess the best one would be page 930. So what you have to understand is this takes place in the mitochondria, and the mitochondria has a double membrane, if you recall your basic cell anatomy. So in that outer membrane, you have some stuff going on, then you have this inner space, and then you have an inner membrane. So that outer membrane is acting like the cell membrane, letting these pyruvate molecules and stuff come in, because that whole citric acid cycle takes place within that that inner area called the matrix. Then you have then you have that inner membrane, which has all of these special membrane proteins, transmembrane proteins throughout them. And what those transmembrane proteins are going to do is help oxidate or reduce, depending on which way you're looking at the process, the NADH and the FADH2. Those NADH and FADH2 comes from the citric acid cycle. So the process of reduction is when you're adding something to the molecule. So reduction takes place during the citric acid cycle and during glycolysis. The NAD molecule is reduced into NADH. However, is that my phone? It is awfully late for someone to be calling. Grr, telemarketers. Sorry. The opposite of reduction is oxidation. Oxidation is when NADH loses the H and goes back to NAD. When FADH2 loses the 2 H's, 2 hydrogens, and is converted into FAD. And you might think, well, why is that important? It all goes back to that creation of ATP. Remember, ATP is made. It's my crazy neighbor. She's calling my cell phone now. She's crazy. Sorry. She feeds the foxes. Totally off topic. Sorry. So, goes back to the creation of ATP from ADP. Remember, ADP plus P turns into ATP when energy is captured from bonds and the breaking down of food molecules. Well, guess what? When NADH is converted into NAD, that bond is broken and energy is released, energy to be captured in ATP. So the NADH and FADH2 that are coming from the citric acid cycle go to this inner mitochondrial membrane and they're oxidized, right? The hydrogens come off of them and that energy is then captured to create a ATP molecule. Now it happens in an interesting way because the way this mitochondria actually works is it's pulling, let me draw this, it's going to make more sense. It's going to pull, use my little eraser sack. This is our outer membrane, this is our inner membrane, So, kind of looks like, I don't know, a hot dog maybe. So our NADH and FADH2 are accumulating in this area. And we have all of these membrane proteins within this inner mitochondrial membrane that basically what they're going to do is they're going to oxidize the NADH and FADH2 and they're going to take the H's off of these and pump them over here. And again, this is closed. So basically you have a hydrogen ion gradient. All of these hydrogens holding back on one side. And this is the process now known as oxidative phosphorylation. Because what's happening is we're oxidizing NADH and FADH2, and now we're going to create this hydrogen ion gradient. And guess what? There's a little pump on one end called ATP synthase. And guess what ATP synthase does? It synthesizes ATP. So these hydrogen ions, if one or two of them seeps through, it's like potential energy being captured. Enough potential energy to take ADP plus P and turn it into ATP. So these hydrogen ions are allowed through the ATP synthase molecule. ADP is converted into ATP. So that's oxidative phosphorylation. And that's how the majority of the ATP is created in carbohydrate metabolism. So that's pretty much the whole little process there. There's one last step, and that is the electron transport chain, which pretty much takes place along with the oxidative phosphorylation. That's why it's usually said electron transport chain / oxidative phosphorylation. And that's because those hydrogens, once they're pumped through, are then going to accumulate on the inside. Pull this back up. Those hydrogens, once they're pumped through, if I just erase that, and we know that that happens, so I won't have to have that written there. So we have ATP being created, but now we're having all of these hydrogens over here. Well, guess what? Oxygen is waiting there for them, and oxygen is going to grab two of these little hydrogens, and guess what? We have a happy little water molecule. Oxygen is the final acceptor of these hydrogen ions at the end of the electron transport chain. And so the end product of carbohydrate metabolism is water. So our products at the end of carbohydrate metabolism aren't just ATP, but we had carbon dioxide that was given off along the way, as well as H2O, as well as water. And that's really the whole process of carbohydrate metabolism. It's all about creating these energy-carrying molecules, NADH and FADH2, so they can go to that special membrane, so that that gradient can be created and hydrogens can be pumped across so that ADP can be converted into ATP. That's the most efficient way the cells have found to create ATP energy. And that's pretty much metabolism in a nutshell. Because what's going to happen with whether it's a lipid or a protein, it's going to be converted into one of those sub-steps along the way so that electron transport chain and oxidative phosphorylation can still occur. So once you understand carbohydrate metabolism and the ins and outs of it, then you're going to understand lipid and protein metabolism as well. So let's refer back to your book. Page 931 does a really good job of showing you how other molecules like triglycerides, lipids, are going to be used in metabolic pathways. And the glycerol and the fatty acids will enter at different parts. So remember, a triglyceride, which is the majority of our fatty products, right? The glycerol is going to be converted into a subsidiary of glycolysis, which is then converted into pyruvic acid, and then it's going to follow the same steps just as if it were a carbohydrate. And then the fatty acids that are floating around in your blood, we call them free fatty acids, FFAs, those float around, they're going to be converted into acetyl-CoA, and they'll jump right into the citric acid cycle, join up with oxaloacetic acid, give off the NADH, FADH2, electron transport chain, etc. The proteins are going to be a little different, and we'll look at those in a minute. But you see on that chart there that some amino acids are going to enter into acetyl-CoA as well. So again, it depends on the amino acid. They're all a little different. And you see that there on page 933, where they all enter at different parts or different points of the cycle. So in other words, glycolysis does not occur with proteins. Protein digestion will occur as normal, right? Proteins are broken down by the peptidases in the stomach, and then the peptidases from the pancreas and the intestine. And then once they're absorbed and they go into the blood, and then they go to where they need to go into the cells, depending on which amino acid we're looking at, it could go into the citric acid cycle as pyruvic acid, or it could come in at any of the other subsidiary steps. But it will ultimately be used to create NADH and FADH2, just like the glucose did in carbohydrate metabolism. So it's just going to depend on which molecule you're talking about. Now, amino acids are going to ultimately have to undergo some modifications, because again, those amino acids that come in from our nutrition are mostly not used for energy. Our body does not like to use proteins for energy, and our body would much rather use proteins, amino acids rather, to build things like our hair, skin, nails, enzymes, etc. And what you see on page 934, the process of transamination and deamination, are metabolic processes for proteins. And I think I'm going to stop here because I'd like to get into that a little more in the next video. So, thanks.